Welcome to the Wright Science Daily Revision Boost. First thing, today's formula is that for density. So density is measured in kilograms per meters cubed, and we work it out by doing the mass in kilograms divided by the volume in meters cubed. In today's biology blast, we're going to have a look at some cells. So hopefully we remember we've got two types of cell: prokaryotic cells, which are the bacterial ones, eukaryotic cells, which are the plant and animal cells. Few key things to remember: prokaryotic cells, no nucleus, they're very simple, and they're smaller, 1 micrometer to 10 micrometers. Eukaryotic cells have a nucleus containing genetic material, they're more complex, and they're larger in size, 10 micrometers to 100 micrometers. Hopefully by now we can draw, label and name all of the key parts of an animal cell. There are only 4 we need to know: the cell membrane, the nucleus, the cytoplasm and the mitochondria. The plant cell, slightly more complex. We've got a couple of extra bits in here. So around the very outside we have the cell wall. Just inside that is the cell membrane. Then we come to our cytoplasm, which has the nucleus, the chloroplasts and the mitochondria in it, and then in the middle usually we have our permanent vacuole. The third type of cell we need to know are the prokaryotic cells, so make sure that you can label these key parts on there. You will have a cell wall, a cell membrane, cytoplasm and genetic material in all prokaryotic cells, but some may also have pili, the slime capsule, plasmids and possibly flagella. Key thing to remember about prokaryotic cells is that they are the bacteria. They're unicellular, one cell. They're the smallest living things because they can carry out the 7 life processes: moving, respiring, they can sense their environments, grow, reproduce, excrete and also take in nutrition. 3 examples of prokaryotic cells on the right for you: Streptomyces, Streptococcus and E. coli. In terms of what our organelles actually do, the nucleus controls the cell and contains genetic material found in all but prokaryotic cells. The cytoplasm is the site of chemical reaction found in them all. The cell membrane controls what enters and leaves the cells and contains receptor molecules found in them all. The mitochondria contain enzymes for respiration and is the site of respiration found in animals and plant cells. The cell wall is made of cellulose in plants, peptidoglycan in prokaryotes, and its purpose is to support the cell, found in all but animal cells. The chloroplasts contain chlorophyll to trap light energy for photosynthesis, only in plants, and the vacuole contains cell sap and helps to support the plant, only in plants. The last 5 organelles we need to think about are ones found only in prokaryotic cells. So the free genetic material is a circular strand of DNA found in the cytoplasm which contains the bacterial genes. The flagella allow the cells to move. Pili allow the cells to attach to surfaces and other cells and also allows them to transfer genetic material between each other. The slime capsule protects the cell and helps it to stick to smooth surfaces. And the plasmid is a circular DNA bit that stores extra genes. If you want to know more about cells, obviously we have our more detailed review which you can click the link for. Don't forget, our daily formula is that for density, and density is measured in kilograms per meters cubed, and we work it out by doing the mass in kilograms divided by the volume in meters cubed. In our chemistry check today, we're going to have a look at the structure of an atom. So first thing, we should be able to reproduce that diagram now. So in the center we have the nucleus, which is made up of the protons and the neutrons, and the electrons are found in shells arranged around the nucleus. We need to know the relative mass and relative charge of our 3 subatomic particles. So a proton, relative mass is 1, relative charge plus 1. Neutrons, relative mass is 1, relative charge 0. electron, relative mass 0.0005, and the relative charge -1. And remember, we use these relative masses because otherwise the numbers we'd be using would be incredibly small and incredibly difficult to use. When you look at the periodic table on your exam, remember each of those boxes contains 4 key bits of information. The atomic number, which is the smaller of the two in the box, the symbol and the name of the element, and the mass number, which is the larger of the two in the box. When we talk about the atomic number, that tells us the number of protons or the number of electrons. The mass number tells us the number of protons plus the number of neutrons. If we talk about an isotope, that's an element with the same number of protons and electrons, but a different number of neutrons. So the atomic number is the same, but the mass number is different. And if we talk about an ion, we are talking about a charged particle. If you're asked to work out the number of subatomic particles, then to work out the number of electrons or protons in an atom, just write down the atomic number from the periodic table. To work out your number of neutrons that are present, you do the mass number minus the atomic number. So when we're talking about ions, which are charged particles, they are formed when we either gain or lose electrons from the atom. If we gain electrons, then the ion will have a negative charge, and if we lose electrons, the ion has a positive charge. If you want to know more about our topic, then go on over to the links below. And of course, our formula for today is density measured in kilograms per meters cubed, which is the mass in kilograms divided by the volume in meters cubed. In our physics prep today, we're going to go over the structure of the atom and how we came up with its model. First scientist, John Dalton, carried out experiments to see how the elements combine, and he came up with the idea that an atom is a very small indestructible sphere. He said that all atoms in an element are the same, and atoms in one element are different from atoms in other elements. The second scientist was J.J. Thomson, and he used cathode rays which were given out by hot metals. And he discovered they're made from electrons, and he worked out that these electrons are negative. So he came up with this idea of the plum pudding model to explain the neutral atom. So he said there's this positive cloud with these negative electrons embedded in it. The third scientist is Ernest Rutherford, who worked with Geiger and Marsden, and he discovered some materials emit positively charged alpha particles. And when he fired those alpha particles at gold foil, then he couldn't explain his results using the plum pudding model. So he said that the atoms have a nucleus which is positively charged and therefore deflected the alpha particles. The fourth and final scientist is Niels Bohr, who suggested that electrons only moved in fixed orbits called electron shells, and this is still the model we use today. Last thing to remember, an atom is about 1 × 10⁻10 meters in diameter, and the radius of the nucleus 1 × 10⁻15 meters. If you want to know more about our structure of the atom, then go on over to the link just below. Now we've had a quick recap, then head on over to socrative.com and test your knowledge on the topics from today. The room name you need to enter is Wright 2662. Good luck!